And are you still at the Zoological Research Museum?
In Bonn.
Okay, great.
Yeah, not yet right now because I'm retired, but that's my affiliation under which I publish. Yes, yes. So I did all the digitization of my tapes and so on. I did that in Bonn. But fortunately, this is now finished because last year, because of the pandemic, the access to the museum was really limited. And fortunately, I finished most of it in 2019. And we had a sound lab at the Museum König, but they closed that down because the Custos was a vertebrate Custos, Gustav Peters, and he retired, and that's why this lab, unfortunately, I think was closed because they thought, okay, we don't need room for tapes. The big player now is Tierstimmenarchiv in Berlin.
Okay.
Yeah.
Yeah, it would be interesting to learn about the different places studying bioacoustics in Germany. Baruch Ghani, who was on the Zoom and who is at our center over the summer, he lives right now, I guess where is that, in Hamburg? But
That's right.
he's been our connection to German bioacoustic projects, at least mine. So should we get started?
Yes. So now, just to finish that, I'm in Alicante in Spain, where I live.
Yeah. Okay, in the very hot Alicante.
Yeah. Okay, so should we start?
Yeah, I'll give just a very small introduction to somebody who spent decades recording the sounds of tropical forests with a concentration on insects in particular, but I think you've studied patterns in biodiversity more generally as well. And I can safely say you've been in this even before, bioacoustics became a very hot and growing field. You were out there with tape decks, making recordings of various species, in the 90s and 2000s, and before we had this, technological revolution. And so it's going to be really interesting to hear about all of your work in this field. And then also, just selfishly as a somebody who works with terrestrial soundscape data, but doesn't know a lot about, insect biology and what we can learn from insects in our passive acoustic monitoring data. I'm very eager to hear your presentation. And so Klaus Riede is joining us now from Spain, but he's a senior scientist at the Zoological Research Museum at Bonn, where he's recently retired, but I think still very engaged with what's going on there. And so, yeah, thank you so much for joining us.
Thank you for the invitation. And I got this invitation from Larissa, Insect Bioacoustics, and I said, wow, that's a big field. But then she asked me along the lines you just outlined to focus a little bit on what maybe vertebrate bioacoustics people should learn on how to avoid clutter, or vice versa, maybe how to share their data so that insect people can appreciate their data and we get a little bit more rapid in insect biodiversity assessment. Okay, so the outline of my talk is some definitions and history of insect bioacoustics, and then an overview about stridulation in insects, with remarks about some of those who made it into model organisms in neuroethology and neurobiology, which has a very strong tradition in Germany and also in the US, of course. And bioacoustics, and finally, and that's where we meet with soundscapes and passive acoustic monitoring, bioacoustics as a tool for species discovery and monitoring. So early milestones in orthoptera bioacoustics were Yersin, who painted scores of insect songs. Then there's another important promoter of insect bioacoustics, which went via the thermometer cricket at the end of the 19th century, which made it into the newspapers and so on, that you could determine the temperature by counting pulses of crickets. And everybody got very enthusiastic about that. And there was a real hype about lots of newspapers, West newspapers, and people started with their stopwatches and so on and counting pulses. And from there on, extrapolate the temperature. That's possible for one species. Okay, then Riede in Germany in the 19-something demonstrated song and phonotaxis in crickets by telephone transmission. So he made a telephone transmission of cricket song and could show that there is phonotaxis of the female. And in the 1930s, we had the technology ready for first sound recordings. Well, in fact, in the 19-something, we already had these Vox tape machines, but I think there are no relevant recordings of insects during that time. So a little bit later in the 1930s, 1940s, the first huge sound recorders appeared. And in the 1950s, analysis equipment appeared, which I will demonstrate now a little bit. And then there's another breakthrough, I would say, in the 1960s, which I mention particularly because you are vertebrate people. So it's Kenneth Riede, which with his seminal experiments about avoidance of bats by moths, which with some spectacular shots illuminating the moths evading the bats. I tried to find this picture, but I didn't find it. And there should also be films. In any case, there's a good review, which I have linked down here. So this is the grasshopper score by Alexander Yersin. There were some more from US scientists a little bit later. If you go in Biodiversity Heritage Library, then you could find them, but I think in this context, this is enough. So what you see here are 30, overall he scored 38 species, even differentiating between closely related species like Cortypus mollis and Cortypus bruneus, and also observing silent stridulation by grasshopper larvae. They move their legs, but of course, because they don't have wings, they don't produce sounds. And what you see here already is it's not so much the pitch, because the grasshoppers produce a noisy sound like shh, but the rhythm is very, very pronounced. So he took some effort to demonstrate this rhythm, and here a crescendo, and here a little bit an impression of a lowering of the pitch. But basically it is the rhythm which is decisive, and it's quite impressive how he managed to capture that with this score. Then my personal hero is Albrecht Faber, who was sort of an outsider, but in any case, he made seminal work in the 1950s, and it is called Sound and Sign Language in Orthoptera. So it's not only sound, it's also sign. And he took great effort to describe meticulously the sequence of movements which are accompanying sound in grasshoppers. There are some spectacular examples. Meanwhile, several of those are in YouTube, fortunately. And also Albrecht Faber made film, and he made film with sound. And we managed to digitize part of its archive and made it available via the Orthoptera species file. But the book is really outstanding because it's so meticulous and still valid, because it describes some species which hence have never been studied anymore. So then we have the studies by Busnel, who already managed again to film. I think they even did high-speed film and synchronize that with oscillograms. So here's, you see the oscillogram of a tedigonid song. Actually, this looks like a today oscillogram, which you produce by all the tools we have now on our PC. Of course, this was a huge apparatus, and you had an oscilloscope, and the oscilloscope was illuminating some light-sensitive film, and later on you had rolls of output of these oscillograms. I still work with those for some years. At the end of my study time in the 1970s, the apparatus was still available. That was the pre-PC time. And they also managed to do some power spectrum. Don't ask me how they did that. They had a special machine for that, and they sort of programmed it, but of course it wasn't a computer. And of course they were well aware that there was a stridulatory file. So here basically you have all together what you need to describe such a tedigonid song. Basically today we don't have much more. We have the power spectrum, which here is going into the ultrasound, and we have the rhythm, and we have the time scale. We have the oscillogram. The spectrogram doesn't bring much more information because we already have the power spectrum here. So that was a great progress, and sort of triggered off a lot of comparative studies. So here you see a very short-winged atypica, a tedigonid, and with very, very short wings, but nevertheless able to produce sound. So here again, the sonograph. This is the sonograph machine. It's basically a filter bank. So here, this cylinder is moving while a tape is rotating in here. You have a 2-second loop of tape which rolls and rolls and rolls, and meanwhile this cylinder walks up, and here you have filter banks, and depending on what is passed through the filter, you get a more intense or more or less intense coloration, and you get an output like this, which basically looks like a sonogram. And I also worked with this, but then of course it became obsolete with the development of bigger machines like MEDAF or so, which were able to do the same thing, but much more elegant, and you don't have to produce the 2-second loop and so on. So this was already a hard and software, which basically does what our PC does now, but of course it was huge. I think I have photographs somewhere before they closed our bioacoustics lab in Bonn, and I think there was still someone standing around there. Okay, anyway, let's stop here. That's history, and we can be happy now that we can do sound analysis everywhere and very cheap, even in the field and so on. Actually, when preparing this talk, I came across this beautiful book, which appeared right now, Exploring Animal Behavior Through Sound. And I found this beautiful picture here of ornithologist Peter Paul Kellogg making recordings of the ivory woodpecker. And big equipment was still needed until recently for ultrasound recording. Ultrasound recording really was a problem. And I think the great breakthrough in ultrasound recording were these passive acoustic recorders. And I'm very happy now with the audio moths, which allows me for less than $100 to record high into the ultrasound with 384 kilohertz sampling rate. So catching all the katydids. So we are improving our data multimedia acoustic database on katydids right now, because what we have in our databases was often based on Racal sound recorders with heavy ultrasound equipment, which of course was only possible in the lab. So publications, where do you publish all this stuff? It started, of course, with vinyl discs, which are still working. That's something I want to see, our digital information, where it is in 70 years from now. Then CDs, the CDs, well, you know, with some of my CDs, I have some difficulties. The tapes are already also still in good shape, but we are now at the critical age where tapes are disintegrating, so they should be digitized. And they are stored in phonotapes. For example, we had several tapes in Bonn, and then we have the big collection of tapes in Berlin. Tierstimmenarchiv, and then we had a lot of private archives, which we were able to bring together during our DOSA digitization project. And this is because many university people were working in neuroethology, hence making recordings. For example, Helversen in Erlangen, he had tapes, Schmidt in Hanover had tapes, and so on. So we brought this together just in time in the 2002 or so, and Klaus Gerhard Heller and Siegfried Ingrisch, they did a lot of work in digitizing that, and we got a huge grant from the Ministry of Research because digitization was just coming up. And then we have the orthoptera species file. I will speak in a minute about that. I just picture of this book, and I just want to say how important books still are, because if you are if you are searching the web for what I'm telling now, for the things, you might get overwhelmed with information and with very sophisticated studies. But basically, you probably get confused as an outsider because, as I show, basically many of the concepts and many of the basic data are still the same as decades ago. It's just getting more refined, And so we have an explosion of publications, but that sort of clutters a little bit the view of the essence. So some books are good, and I will show you some in the course of the talk. So this is Orthoptera species file. Unfortunately, it is a taxonomic database, so it is focusing on literature data, where have species been described and so on, but it also has a lot of pictures of museum specimens, but also of live specimens. And we started to put our sound recordings from the DORSA project into Orthoptera species file, because we had the severe issues with the university server, which was in Ulm. And fortunately, we started to migrate the data. The migration isn't complete, and still the database design for multimedia in Orthoptera species file is not optimal. But we are working on that, and we hope that we can sort of save our DORSA data, which are like of 6,000 sounds files from 621 species. And meanwhile, filling and populating the OS Orthoptera species database goes on. The good thing about Orthoptera species database is that it is always the latest taxonomy. So they are doing continuous monitoring of new description of species. And one of the main problems we have in insect bioacoustics, if we go beyond the model organisms and beyond the well-known European and North American species, we need voucher specimens. And we need an association between the voucher specimen and the sound, because in many cases, the species is hitherto undescribed. Okay. So what are we talking about? We are talking about frequency ranges from very low infrasound up to 100 kilohertz, so high into the ultrasound. And here is, let's say, the sound perception space of a cricket, which is perceiving with tympana in the front legs, and with cerci, with small hairs, wind-sensitive hairs, which are going down into the infrasound. So the infrasound is needed for predator avoidance, like, for example, this is another neuroethology paradigm. The toad, which is trying to catch the cockroach also has these hairs and makes an evasive flight response. And then you have, of course, the bats, which are perceived by these tympanal organs. Both these cockroach work as and the bat avoidance were studied in, I think, even in New York. Ron Hoy, do you know him? Maybe he studied those, and then there is this work about the giant interneurons detecting the wind. Okay, so I hope, do you see this side of the screen here? Here, and I hope the sound up there doesn't disturb too much. Here I combined an evolutionary theory of insects with those groups which are able to produce sounds. You see, it's not only Orthoptera, and it's not only Cicada, it's also Lepidoptera and Hymenoptera and Diptera, and beetles, of course, Hemiptera. I can't speak about all those, of course, today, and I'm not a specialist. I'm mainly with Orthoptera and a little bit about Cicada, but I want to mention them because many of you are starting their career, and I would say that most of these other groups are under-researched. So there is a lot in moths, for example. There is a lot to do. And for example, we have this audio moth, but up to now, I couldn't identify a moth click, which evidently is somewhere out there, maybe for predator interference. So there is a lot of work to do out there, and I want to recommend this book here, Insect Sound and Communication, and I will provide the link when I make the version to load up somewhere. Okay, so I want to present, before I start with the Orthoptera, one example from Diptera of a model organism, and this is Drosophila. Drosophila has a male courtship song, it has a female copulation song, it has sexual rejection sound, aggression sound. You see all characterized by pulse structure generated by the wings. Now, don't worry, they won't clutter your tape recordings, because these sounds are very soft and they are working in the near field. So here we have sound velocity and in the near field. And they have become a model organism for genetics, neurogenetics, and the advances are really surprising, going deep into the neurobiology and deep into the control of certain neurons by certain genes. Fascinating stuff. Hearing in Diptera, for example, mosquitoes, is by the antenna, but by Johnston's organ in the male. You see here this bushy antennae by the male, which don't hurt you. The females are blood-sucking. And the reception of the sound is down here by scolopedial organs. And then we have the moths. The moths do have a tympanal organ here, you see in the abdomen, and different moth families have the tympana in different places of their body. And about the sound production mechanism, this is a little bit messy. And as said, in these moths, we have sound signals directed at a predator, and we have alarm signal directed as a non-predator, and so on. So we have sort of an acoustic aposematism, and of course, via the tympana, moths can perceive bats, and then make this famous avoidance maneuver, which was documented by Kenneth Riede. But as said, it's totally under-researched. So this here is from Jack, which she made a nice review, which is linked here. And she's working on that, but I think she's the only one who has an overview. And yeah, maybe you can invite her to your seminar. Then sound production in cicadas. This is a different mechanism. It's a click mechanism. It's like if you have a Coca-Cola tin and you press it, you make a little sound click. And this sound click mechanism is produced by big muscles here. Here you see the different patterns. You see impressive modulation of frequency, and you see impressive bands, which are not harmonics. These are not harmonics, these are formants. And that means that a cicada song could sound like wah, 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 and can be described by a human vocal, actually. So a totally different mechanism from as we have now in Ensiphera and Sellifera in Orthoptera. We have katydids and bush crickets, which are Ensiphera, which are these ones here, and which you can recognize by their long antennae. And mainly we are speaking in the following about Tetigoniidae, even though you have sound production also in Grillacrididae and Raphidophoridae, Schizodactylidae. Again, a huge field, especially in those one here. Tetigoniidae is well researched, but the other ones here, tropical families, terra incognita, I would say. And then we have the Acrididae, the Sellifera, again, mainly in the Acrididae, we have sound production. And yes, so I'm an acridid specialist, so now you will hear a little bit more about acridids. So the basic sound production mechanisms in all Orthoptera is a file and scraper mechanism. So you have two wings here, which are sort of asymmetrical. One wing has this stridulatory file on the underside, this plectrum, and the other has a scraper. And by moving both against each other at high speed and with a certain rhythm, a specialized region here on the wing called the harp is radiating a sound. Generally, insects have a problem because they are so small, and the smaller you are, the smaller has to be the wavelength you radiate. But as you know, crickets are well in the audible, and some are using tricks like baffles or leaves to increase the radiation surface for their sound. I won't go into that. This alone is a super interesting theme, and I have suggested to Larissa to invite Fernando Montealegre, who is still continuing this line of work, which has been abandoned, and he took it up again and is doing now very sophisticated work with laser vibrometry to find out which regions of the wing exactly vibrate and so on. And in Acrididae, you have a different mechanism. It's basically a row of pegs at the inside of the hind leg moving against the hind wing. You see here against the tegmina. So these are moved against each other, and sound is produced. Sorry. There are some other mechanisms, stridulation by wings in Romalaidae, which are the lubber grasshoppers, which you have in the US. And they are producing very audible sounds. And there's also a very interesting species in South America of this group of lubber grasshoppers who produce their sound by opening their wings and closing them, which sounds like a bird. So if you go into iNaturalist and listen into the sound recordings, filter by sound recordings, then search for Prionatris. These are the ones which are sounding like birds. So I'm quite sure if you put that song as a mystery song into Xenocanto, then interesting guesses will come up. I wanted to do that all the time, but never did it. So maybe one of you is taking up this idea and doing that. Okay, so you see here rows of little pegs. So basically it's always a scraper against a plectrum, like moving a stick against a fence. That's the basic mechanisms. And then the sound is produced. Here is this hindwing resonator, which is also very nice because this Hyalopteryx species is producing is singing on the wing by moving, fluttering, which is wing, and due to these resonance structures, you have either very short clicks, or in this case here, a ring-like tone. So very interesting. I'm working right now about that. And it's not quite clear how the sound is produced here. It's probably like moving these veins against each other, and from there on, from the disbalances, stimulating these thin area in this case, on the hind wing. And below is the tympanal organ. You see here is the tympanal organ of the Riede, of the short-haired grasshoppers, which is in the first abdominal segment. Okay. So here's again the tympanal organ, and this is from 1906, from Schwabe 1906, which already elucidated all the structure and so on by microscopic dissection. Müller's organ, Müller, as you find, is a German name. They did this in the 19-something, end of the 18th, beginning of the 19th century. And here I won't go into detail, but these are the grasshopper families which are singing versus the ones which are not singing. A lot are not singing. So here, the taxonomic value of songs, you see very closely looking grasshoppers from the Cortipus group, which are very well differentiated by the distinct temporal patterns of their songs. So that's the great value of grasshopper songs and cricket songs for taxonomy. And finally, I wanted to comment a little bit of history again, and why we have so many recordings in Germany, because there was a big school of neuroethology by von Helversen and by my thesis supervisor, Franz Huber, who wrote this book about cricket behavior and neurobiology, and the Helversens about neural basis of behavioral adaptations. And Helversen developed a method to monitor the leg movements and the sound. And you see here, there's an optoelectronic device recording the leg movement, and here you have the sound output. So we exactly know how each movement of the legs and each position is producing which song and its behavioral function, etc. So the whole chain from the sound production to the brain has been elucidated in both sides. So muscle control, but also reception. So here you see this optoelectronic device to monitor the position of the leg, here the microphone, and here in this case, the song has been electrically elicited, and they wanted to study in these studies the muscle activity which is controlling the song. This, I would say, is a closed shop, because sort of everything of this chain is known. I think it doesn't make much sense to go deeper into that. So here again, not only sound, but also movements. So there are always some species in each community which are producing very conspicuous dances. In the US, it's a Zurula species. In South America, it's, for example, Peruvia nigromarginata. You see they have a nice white antenna, and they use it from their producing of a dance with these antennae. And then there's the one which I have studied in Germany, which is Gomphosorhipus rufus from Eurasia, which is also producing a very complex dance in front of the female plus a sound. So in fact, we don't need only sound databases, we need movie databases to get all the information. We have YouTube, but I would say as scientists and museum people, we can't rely on YouTube to store our data. So here I leave that away. I think it's just the tympanum inside and the sound reception range. It's going up to 40 kilohertz. And now that said, as I said, the high ultrasound component of insect sounds, especially from katydids, which go up to 120 kilohertz in the South American jungle, require that we record with high sampling rates. MP3 is by no means enough. So here you see the MP3. This is this violet line power spectrum against a RAF file, which covers the entire spectrum, I think in this case up to 40 kilohertz. And you see that the MP3, of course, is cut. So you are only covering a part of the spectrum. In grasshoppers, that works, but in katydids, in most cases, it doesn't work. So you don't get them with an MP3 if you are targeting them. And if you have MP3 files from soundscapes, it might be problematic to use them. But if you want to study rhythm, like cricket rhythm of their songs, then it's sufficient. So here's a nice paper by Filippo Buzzetti and Pavan, which make a plea for using wideband sound recorders for field investigation. That's easy to say because now we have these cheap machines. And I would say a decade ago, it was impractical. Only very few people did ultrasound recording in Orthoptera. So now I come to the soundscapes and my idea of using bioacoustic monitoring for assessing insect diversity in tropical forests. So as you all know, insects make the bulk of diversity, especially Coleoptera, and guesstimates of species on Earth. You remember this interesting discussion between Irving and Stork. Irving 30 million, Stork topped that, 80 million species on Earth based on canopy fogging data and extrapolations. So there was a huge discussion going on, but I think everybody is zooming in now into sort of 5 million species. But how can insect diversity be mapped or monitored if most species don't even have a name? So I thought that bioacoustic recording and classification of Orthoptera is a non-invasive rapid assessment of species communities, particularly in species-rich tropical forest habitats. And what you can do is monitor species presence absence. You can monitor species ranges and detect endemisms. So that would be species discovery. If I record a sound which is not yet in my database, it's probably a new species, maybe a new species, and I have to go on searching. And on a regular basis, you can do species monitoring, species abundance throughout the year or during recovery of ecosystems, activity patterns, community patterns, extirpation rates, and species recovery. So acoustic profiles of Orthoptera communities, however, are rare. So we have some of my work here and Nish, my doctoral student, which we did in Ecuador. Then we have the cricket recordings in North America by Walker, which are all databased in the SINA, the singing insect of North America database. Great work. Then we have the work from Otte and Alexander in Australia, but this is published as a book. Then we have the group of Balakrishnan in India. And then we have the whole bunch of people who are doing passive acoustic monitoring and establishing bioacoustic indices, acoustic entropy. And of course, if you look into these bioacoustic diversity indices, you see that many of them are driven by insects. For example, this seminal work here by Sir, by Acoustic Entropy in Tanzanian Forest, where he has introduced his bioacoustic diversity index. And but of course, this is general, and it doesn't go into the single sound tracks. So, so let's have a look into these, for example, these old work here from Australia, which is published as a book. There should be some tapes around somewhere. And there are these songs without a voucher. So there should be a species out there which has not yet been collected. The same by the work from Holger Braun in Loja, Ecuador, 25 songs, which are characteristic song patterns, but without a voucher. So still much work to collect them. And finally, again, our work in Panama with crickets, 13 crickets songs without a voucher, which we still have not yet collected. But even if we have the collected specimen, it doesn't help us much, because now the whole taxonomy has to start. So I call these specimens, which don't have a scientific name, types of tomorrow, because they will be the holotypes for the hopefully described species in the future. But we have everything there. We have the specimen, we have the song, and so for future taxonomists, it should be possible to describe the species, and hopefully it will also exist in the wild, continue to exist. So let's go to some of the soundscapes. Here I have featured some distinct places in distinct part of the world. So this is the yellow one here in the Italian Alps. This was only a snapshot, habitat to similar where I'm now, Mediterranean. Now here, this is the Ecuadorian rainforest, where I've done quite a lot of recording. And here is Panama, where we have done this more sophisticated work, including ultrasound work, which I will comment in a minute. And finally, we have my seminal work, which Ben mentioned in the introduction, in the 1990s in Borneo, where I used cassettes and an array of microphones to do simultaneous recordings of a soundscape in the canopy of a Malaysian rainforest in Borneo, Sabah, Borneo. So let's have a look into the soundscapes. Here the Italian soundscapes, which was rather quiet, no cicada at this time of the year, but very nice song of an acridid, of a shorthorn grasshopper. Rio Agua Rica, Ecuador, with a dense band of crickets and frogs. And this doesn't go up high in the ultrasound, otherwise we would see a little bit more here. And the Barro Colorado soundscape looks similar, with this, again, with this band here, which is produced by very small crickets between 8 and 5 kilohertz. And finally, pouring Sabah, Malaysia, which sounds quite different. And this is dominated by cicadas and also some Telegonidae, but which are more sounding like frogs. So already visually you see that the sound is quite distinct. So this was my arrangement for the rainfall for the canopy in Sabah. So we had sort of a canopy walkway. This was the experimental one, work in progress. And you see here's a microphone hidden. These were electret microphones, and I connected with 50-meter cables to mixers, and so I could select the channel. And so I went in 5 minutes interval from microphone to microphone and taped all this on cassette. And these are the small electret microphones. Okay, so this was this, and fortunately we have digitized great part of it, and we also have elaborated at least the cicadas, which were dominant, as said. So we found out this special dusk chorus of cicadas, but still there's some work to do. There are still some species which we can't coordinate with the respective voucher specimen. And 30 years after the vocal recording of katydid, we finally managed to describe the species. So as said, we had the song, we had the recording, we had the specimen, and thanks to iNaturalist, we also had some Siegfried English got enthusiastic when he saw these photographs here from closely related species, also from Borneo. So we described this bizarre pink katydid from Borneo, and yeah, this is the ideal case, the voucher specimen and the sound and stridulatory file. Maybe it would be good to have the genetics and then the song available in a publicly accessible database. So that's the ideal case. We just published another paper on the Mecopoda, so you see how important it is to keep these voucher specimens safely in museums and to maintain the association between sound recording and specimen. How much time have I got more?
So I was just going to write a message about that. Usually we end at 11:30, so in 5 minutes. But
okay.
I think, especially because you're getting to topics related to the soundscape and all of this is very interesting. I think that, you know, we could go longer if you still have slides that you'd like to present on this topic. And we also have some questions that I'd love to get your thoughts on with regards to, I don't know, the future of describing insects using acoustic monitoring and just various questions. So I hope that we have time for that as well. Are you able to stay a little bit longer?
Yes. I have no problem. Okay, so I go and I think there are 10 minutes more and then I'm done. Okay, so here now our Panama. The Panama station, maybe some of you know BCI, Barrow, Colorado Island, managed by Smithsonian, of course, is a gem in tropical, it's a mecca of tropical biology research. I have the impression that about 30% of the publications about tropical ecology and tropical organisms are in some way or the other made in Barrow, Colorado Island. And particularly what was fascinating for us as neuroethologists trying to combine sound production and nervous perception, was the availability of labs. So we had lab space there, and here you see Arne Schmidt, who was a doctoral student, and Anna Einhobel, who helped us. And this is Heiner Römer, who is a very famous neuroethologist because he developed the inter alia, because he developed the biological microphone. So this is mainly making nervous recordings in the field. So you have a katydid, and you have electrodes attached to their ears, and so you can record actually what the katydid is hearing in all this mess of sound. And so, but that was not exactly our topic, and I'm coming back to what Ben just mentioned. So these are the soundscapes here, which I already showed, and here a little bit clearer, you see already distinct patterns of crickets characterized by their distinct rhythms, and you see here some higher pitched katydids. So now you apply filters, and you can filter out, for example, this 6 kilohertz cricket. This is one way of isolating species. The other way is focal recording, where you go around, as you all know, with a parabole and record species. And the third is to make your acoustic library to record crickets in captivity. So we collected crickets at light or with pitfall traps and had them in the lab and made the recordings. And then we had everything, the recording and the voucher specimen, etc. So we could establish a sound library of the species occurring at this place. And I think this is necessary because probably a species singing with these acoustic parameters here, maybe you find a similar species in Amazonia or even in Borneo, but this will definitely be a different one from the Panamanian one. So each community should be studied on its own, and you should have this library and reference data set. And yesterday, a colleague here from Alicante asked me, so how much is the distance? How far do you have to go to find new endemisms? And I said, I think simply don't know. And I think in Panama, for example, with this humidity gradient, in 100 kilometers, maybe you already find another community of crickets. And even here in Alicante, with all its mountains and valleys and so on, you probably have a lot of micro endemisms. And so we did this work, which we published in some neuroethology and so on, but still there are a lot of species which don't have a scientific name, and I didn't have the nerves to go deep into the taxonomy. It was really difficult. You need a very good taxonomist to do the work. So Paris has a group, Desouté, etc., and Tony Robilia, who are doing that. But as said, this for us, as let's say from an ecology or ethology point of view, it's enough to have this ethos species. And we give it a preliminary name, and we even can database the preliminary name, and that's it for our studies. And with katydids, it looks a little bit better. And just yesterday or so, a paper appeared about the katydid calling activity from soundscape recordings in Panama. So finally, some sounds, and I didn't write to those, so maybe now you are able to guess what is what. guesses?
Diego says cicada, and I think it might be from Borneo.
Yes, yes. It's Pomponia imperatoria from Borneo. It's a dominant species in the evening chorus. Yes, great.
Let's
So
see if the acoustic methods audience, or let's say if we can be undefeated in this quiz, we can mine the collective knowledge.
yeah, no, that was great.
Nice. Okay, so it looks like we think it's a cricket.
Yes.
Potentially from, is it from Tanzania, or where's the cricket from?
Alicante.
Ah, okay.
And it could also be from the US. These are called tree crickets, Eucantine, and they all sound like this. And you can go into the parameters, and they also look like very much. They are sort of transparent wings. So yes, these are the tree crickets. These are the same.
Are those tree crickets?
These are the tree crickets, and I played this to show that there are several of them. You hear that the tone is a little
If you
bit
pause it,
different.
we can hear you better. There we go. Or if you can pause it, we could hear you better. There we go. Almost done. There we
Okay.
go.
Yeah. I played this one because you heard different individuals with different frequencies and different intensities. So these were made with the audio moth recorder right 10 minutes from our city home here in Alicante and some badlands. And they're there in the evening. And a day you see nothing. You see nothing of these crickets. And yes, and even at night it's very difficult because they are so transparent. But with the audio moth, you capture tons of them. And as said, there are several of those with different frequencies, and you see how they answer each other. So even from the soundscape recordings, you can do a little bit of a density estimate.
So I'm wondering if we should get to some questions, because I know that some people have were eager to speak with you. I didn't even realize, I'll just keep this very short, but you know, your 1993 paper, Larissa just put that paper in the chat, but we can send it again. You were one of the first people to really think about acoustically monitoring biodiversity within, especially in tropical forests. And so it's just, again, it's so cool to be speaking with you. And if anybody has any questions about insect sounds in passive acoustic monitoring data. Let's have a good discussion.
Yes. So I will open up this window so I can see more of you. Okay. And you are selecting the questions. I don't see all the hands and so on.
Okay.
I have a question. Ben?
Yeah, Baruj.
Hi.
Sure.
First of all, thank you so much for your talk. It was very insightful, especially for people who have a background in terrestrial bioacoustics here. So it's a very general question. We have all heard reports about that suggest that the abundance of insects is in drastic decline. Which is quite worrying. So what would you say about the advances in machine learning and deep learning? And what could these tools provide us? Like, how could they help providing potential new solutions to this global challenge? So I would like you to contrast it with the terrestrial taxonomy. So what would be the challenges that we would face with, in contrast to, for instance, birds or other mammals like marine mammal bioacoustics? So when we would exploit machine learning tools to do passive acoustic monitoring.
Yes, actually, we did quite a lot of work about machine learning, and I left that out finally. I first wanted to put it into the talk, but it was definitely too long. To make a long story short, the basic problem with machine learning is that even more than by human identification, you need reference recordings. So even a machine can't help you if you only have a voucher specimen. No, if you have a voucher specimen and reference recording, it's good. Machine learning, for example, could help you to make these libraries faster. So it could, all the work which I showed you, which we've done by hand, like filtering and so on, you could do unsupervised learning, and you could say, okay, here are tons of recordings from the forest in India or in anywhere. Machine, please tell me how many different patterns you can recognize. So now you get a bunch of files due to machine classification, unsupervised. You go into that, and then you see, aha, there's a bird. We know that. That's for the birders. These are the insects. There are 10 of those, and so on. So at least you get the mechanic work done more quickly. But this doesn't save you from carefully collecting and from focal recording as we did it in Panama. Somebody has to do that. But anyway, it's great to make rapid assessments. It's great to make rapid assessments. And I'm a little bit unhappy, I must say, with the bioacoustics indices, because the bioacoustic indices don't tell me much about how many insects are there. I would rather have a machine run over these recordings and say, how many insects do you think are in there? Or you could also probably program easily the machines to differentiate what we just did, a cricket from a cicada and a katydid, etc. I think that's an easy task. And maybe there's some confusion between a frog and a cricket, and maybe between some bird and a grasshopper, but this is marginal. So it would be great to accelerate our work, but we do need the underlying databases.
Yeah, thank you so much.
one question. Oh, Diego, do you want to read your question, or I can also read it if you like?
Yes, I can. It's a little bit noisy around here, but yeah. Thank you for the talk. It was super insightful. I enjoyed it very much, especially the historical summary. It was super interesting. Um, what do you think about automatic species identification of insects? I know a lot of work has yet to be done in that regard. I've been researching about it, and I see most of the species identification models are mainly used with birds and frogs, but not a lot of work on insects. So do you think they may be useful for the future of acoustic monitoring of insects?
Yes, definitely. Of course. Yes, yes, yes. I would say the underlying technology of feature extraction, etc., is basically the same. You just have another range of frequency. You are going more into the ultrasonic. What you never will manage is to do machine identification of MP3 sounds of katydids which are singing above 20 kilohertz because it's not in the signal. But if the signal is good and you run the identification, and then of course it should work as well as for birds. You have the training data sets, and you have the recursive networks or whatever, and you can do it. And for example, I'm working a little bit with BirdNet now to identify the birds which are around here, and it works so nice. So I think it also should work for insects. But you just need the training data, and we do have the training data for the European ones. We do have the training data. So it could be done, yes. The problem will be probably the user interface, because as said, the user is recording an MP3, so how can he catch the katydid, which is singing the ultrasound? So maybe he has to connect to a bed detector. So it's more on this side. But from the machine learning side, this will all be there in 5 years. We have that perfect. The problem is the input side, garbage in, garbage out. You see, if we don't have nice reference data, we are lost. But still, maybe we can program a machine learning program which can say, okay, I as a machine, I identify 20 orthoptera in this soundscape. That would already be useful for rapid assessments.
So we've got a question from Tiago. How do you see future work with insect acoustics, with songs now being available in something like a Xenocanto database? And then following up on that, I'm wondering if you see like a time in the future, let's say it's 20 years or 50 years, do you think we'll know the majority of sound, like if we'll be able to identify the majority of sounds to insect species? And what is your time frame for when you see that happening? Is that like where we should be pushing? So first is, do you see, how do you see the future of working with insect sounds? Is this going to be in some sort of database like in Xenocanto? And then what do you see the process for continuing to ID insect sounds in the future? Where are we going?
Yeah, maybe I start with the last one. The idea of insect sounds, of course, is closely connected to the taxonomic impediment. So you need close cooperation with taxonomists to do the taxonomist proper. Even numbering the species doesn't help too much because you have to go into the details. So you see my colleagues now, Hella and Ingrid, are even older than I am. So they are in Brazil, there's a good taxonomy tradition now. And so I hope that from there, from southern countries, we will get good taxonomists to do that. And then the other point are the databases and xenocanthus, etc. That's soft spot, I would say. 20 years ago, I was more enthusiastic than I'm now, because we made this DORSA database and we had the server in Ulm and we had 6,000 songs and it all worked on the internet. And you know what happened? The guy who managed this server died last year. And the successor in the department decided not to continue these servers. So long-term archiving is crucial for all this. And it cannot be done at a university, because there comes another professor and he said, oh, what is this machine over there? Let's save energy and switch it off. So I think the only institutions which can do a solid archiving are the museums, long-term institutions. The museums do a good job for archiving specimens, but they still do a lousy job in digital processing and digital archiving. Digital long-term archiving is I would say, generally an unsolved problem. So this has to be solved. Otherwise, we will always start from the beginning. And that's where I am with Xenocanto. We recently had a meeting with the European orthopterists, and Xenocanto was demonstrated, and it's a great tool. But in a way, they presented it as if they were starting with zero. So they were ignoring everything what we have done. It's amazing. Particularly because they were, up to now, they were concentrating on MP3. So now they are allowing WAV files, that's good. But still in Xenocanto, the link to the voucher specimen is a bit missing. We have this in Orthoptera species file, but we don't have it in Xenocanto. And actually, it's not very necessary in Europe. But if you want to go to the tropics, you need the voucher specimens and you need this link. So the cooperation between museums and databases and long-term archiving is crucial. If we manage, maybe with your help, to push that, then I see a great future. And otherwise, the future is bleak. The internet will just disintegrate and we always will forget what already has been done. And yeah, so that's my prediction. I think if everything works fine in 20 years, we are good at automatic recognizing and databases and so on. Otherwise, we will get lost. And finally, we have to save the rainforest. Don't forget that. Otherwise, what do you want to record?
Does anybody have any questions? I have one more in the chamber ready. So there's a lot of work coming out that's using acoustic monitoring recordings to learn about habitat condition or the effects of restoration or disturbance. And especially if they're using not necessarily acoustic indices, but in many cases they are using some type of acoustic indices, or let's say measuring the power in different band levels. Insects are, an outsized contributor to the soundscape, and yet they're oftentimes the taxonomic group that, at least in my experience, that I have the least amount of ecological knowledge about these different insects. And so what I'm wondering is, you know, if we're trying to learn about, let's say, restoration outcomes, and we have acoustic monitoring recordings for multiple sites, beyond just doing these sort of broad sweeping measurements, do you have any recommendations? Should we focus on like the most obvious or most common sound producers? Should we take time to do some research into what might be like disturbance sensitive insect species and look for, should we pour through the archives? What are some strategies? Because I know a lot of us here are trying to use acoustic monitoring recordings to learn about land use effects and the like. So what would be a good roadmap for how we can more intelligently get the most out of insect sounds in this respect?
Actually, I have a talk about that. As you say, selecting sensitive species or some sort of flagship species or critically endangered species would be a good strategy. For example, there is a bandwing grasshopper in California, which is producing a crackling sound. And of course, this is critically endangered. Trimerotrope is something. I have the slide, but let's leave it there. And so the Alliance of Zero Extinction even has this species on its list. So we know where it is. We know the sounds it produces. So a monitoring program sort of focusing on this insect would be really useful. And the correlation with vertebrates, I don't know, but I'm sure that there are many correlations. I know of one work, I've forgotten who did that, and this was in the context of insect decline, which you mentioned, the worrying insect decline, particularly in industrialized countries due to pesticides, I would say. And this was correlating the abundance of certain birds predating on big insects with Orthoptera. And Pterygonidae are like this and fat, and they are prey of many birds. And he could show that the decline of these big insects was going hand in hand with the decline. I think it was a certain falcon, a small falcon, which was declining. So yes, of course, focusing on certain target species would be excellent. And there's another work by Google about restoration ecology, what you mentioned. And this was an island in Mauritius, and they eradicated the invasive plant species and they planted the original plant species, and they used the small cricket as an indicator species, and the small cricket is indicative of the native forest. So you could use that and with sound recorders, and these sound recording can be done by park personnel, by rangers. You just put the sound recorder there, and in weekly rhythm, researchers go there, or you even can send the chips by mail. You see the stored chips. And so there are a lot of possibilities to facilitate, streamline this monitoring and even automatize it.
I'd just like to mention that it seems that as important as it is to get more people also invested in becoming specialists in the taxonomy of and systematics of these species, meaning it's again bringing more from what has been done in the past in terms of biological studies, it's also interesting to get people to also understand the behavior and the natural history of these organisms, so we can then understand the particularities and their ecology and what should be targeted at monitoring, maybe in a more effective way rather than investigating communities or not.
Yeah, absolutely, absolutely. And that's my personal interest still, because, okay, it's nice to have the name, it's nice to have this song, and it's nice to have these numbers and data, but actually what I'm interested in are the behaviors and strategies and so on, which are so different, and what are they actually doing? And for example, take North American grasshoppers. We have the work by Otte, who did sonograms, and amazingly, we don't have recordings of North American grasshoppers available, and even the behavioral studies are completely lagging. It got out of fashion. We have lots of crickets and katydids due to Tom Walker and due to the neuroorthologists, but these behaviors of grasshoppers, which are flying around in the prairie and fluttering and doing dances and so on, fascinating. At the moment, you find more on YouTube than in the scientific literature, because people are fascinating, they are filming it, and but we don't have it in the databases, because so beyond the acoustic database, we need movie databases, well graded movie databases, and where we document these behaviors.
And then if we're interested in learning more about community biodiversity patterns, let's say, or at least of acoustically active species, even if we don't know the majority of insect species and we don't have a connection with a really good taxonomist or the ability to do the necessary field work, would you still say that it's of value to produce these sort of what did you call them, like sonotypes or ethosounds?
Sonotypes or ethospecies.
Ethospecies. So if we have a acoustic monitoring data set, what's the value and what can we do with that ethospecies list of those different insect sounds that we've found in our data?
Well, for example, you could do mapping, because you are recording at distinct sites, and maybe you detect a really conspicuous insect song there, and you can identify it. I still have some from Uruguay, which I haven't collected, but they are well identified, and you can map it. So we can map the unknown, but you know, if soundscape recordings are well stored, then it should be possible, ideally it should be possible to work the taxonomy out decades later. But still you can publish a work about a certain species which you found always in this habitat associated with, I don't know what, bog. And so, of course, that's extremely valuable. The point is that these data should be shared, these audioscapes. I will add, when I put my talk into the web, then I will add some more slides going into the direction of your question. And again, I have a lot of examples from nice databases of soundscapes which were switched off now. It's also a question of cost, of course. So it has to be big institutions, and maybe we have to think of, not of tapes, but of ways to store digital information independent from electricity and from hard disks. You know, the famous Kubrick little hologram crystal, something like that. passive storage of data. That's I think that will come in the future. So but then it for ecologists it's absolutely valuable, I would say.
Maybe we've got one last question. Jose, would you? So Jose is wondering, what alternatives would there be to think of in cases where Orthoptera overlap in the frequency of other vertebrates, so that maybe I'm reading, like, what do you do in the case that there's many insect sounds that overlap? Jose, is that your question? Let's see.
I think he might be meaning that is this overlap on purpose? Does it say something about if they are competing for the niche?
Yeah.
Yeah.
I mean, so that and then if you have any thoughts on the acoustic niche hypothesis in the context of that question, so
there are some works about that, and actually we did some work about that in Panama, but you know, insects have been first, so long before the vertebrates, vocalizing vertebrates. So maybe there's some direct input, like loud vertebrate calling, sort of interrupting the insect, but basically I would say these are two different worlds. So there is few overlaps. I would rather say inside the insect world, for example, the cicada, which are so active here in during some part of the day or in some part of the season, the cicadas are really sort of stopping other acoustic activity, but vertebrates, I think, are no problem. Even traffic noise, there are some work about traffic noise and shifting in frequencies in insects. I don't see much effect. Bats are a thing. So if you have a bat, I have several recordings where I have the impression that the bat is coming and the katydid is stopping. So yeah, that's an interaction, but it's in the ultrasound. Okay. So I didn't look into the chat, I must admit, because I was busy with the talk. So now I will type in my suggestion where to put the talk, and it's www.de. So that will be the directory where I put up the stuff. And
perfect.
Then we stay in touch, I hope.
Yeah, sure. We can definitely share the link of the talk for those that are interested in seeing your talk. And there are people that already asked for the link to your recording later, to see later. And we'll share the link together of the presentation itself. And I appreciate it, Klaus, for you having the time and taking the time actually to join us today in this talk. I hope everyone enjoy it. And we keep the talk, of course.
Okay.
Thanks so much, Klaus.
The chat.
Thank you.
So thanks a lot. Goodbye.
Goodbye.
Bye-bye. Take care.